Hello, my name is Dr. Claire Gamlin, and I am a scientist on the Neuroanatomy team here at the Allen Institute for Brain Science. And today I will be talking about neurons beyond the textbook, a lesson plan, and I'm going to first provide a brief overview of the Allen Institute and then discuss morphology and why it's important. So here's the overview for today's presentation, and I will cover these first 2 points. So the Allen Institute is a biological sciences nonprofit located in Seattle, Washington. At the Allen Institute, we study neuroscience in the Allen Institute for Brain Science, Allen Institute for Neural Dynamics, and the MindScope program. We study cell biology at the Allen Institute for Cell Science and immunology at the Allen Institute for Immunology. We also support cutting-edge research across the world with the Paul G. Allen Frontiers Group. And across our institute, our scientific process is focused on tackling hard, complex, foundational biology. And our approach is guided by 3 principles: big science, team science, and open science. The most relevant of which today is open science. Our work generates data, knowledge, and analysis tools, all of which we share publicly. And as of fall 2020, we have collected about 14 petabytes of data. That's about 2.8 million DVDs. So our core 3 principles guide all of the work that we do. Team science and big science mean exactly what they sound like. And we also are guided by open science. And that means that we share all of our resources, data analysis tools, products, and our own research findings for anyone to use in their work, including for educational purposes. And this is what makes the Allen Institute truly unique. We have been publishing our data online since the inception of the Allen Institute back in 2003. And this is something that's become really important to our field, and an NIH initiative has been dedicated to it. The Allen Institute is both very involved and a leader in that effort. So we not only share our findings through scientific publications, but we also make our data, analysis software, and biomaterials available to the public. And as I mentioned, this is what makes the Allen Institute different from other scientific institutions. Our online resources, some of which are shown here, include a map of gene expression in the adult and developing mouse brain, as well as the mouse spinal cord, and even human brain. And we've also created anatomical atlases for each of these data sets, and people now regularly map their mouse brain image data to our 3D atlases. We've also developed a cell types database that contains morphological, electrophysiological, and transcriptomic information from single cells. And it's these data that our lesson plan focuses on. So now I want to talk about what is morphology and why it's important. What do I mean when I say morphology? Well, I mean the shape of a neuron and describing where the cell extends its processes, such as the axon and dendrite, as well as the soma location. And understanding, how neuroscientists use morphology to describe cells is really at the heart of this lesson plan. And so here I'm indicating on this drawing to the right, dendrites in blue, soma locations in black, and axons in red. And we care about morphology because we know from the morphology of a neuron where a cell is receiving input, so where its dendrites are located, and where it's providing output, where its axon goes. And scientists have been observing neuromorphology for over 100 years. So this drawing here to the right is from Ramon y Cajal, drawn in the early 1900s. And these are retinal ganglion cells from the lizard retina. So morphology also refers to the nature of a neuron's processes. So whether they're smooth or spiny or highly branched. And you can see here to the right that even though these are all retinal ganglion cells, they do vary, especially in how their dendrites are elaborating. And we also know that neuron morphology varies across brain regions and even within brain regions. So as I'm showing here, even within a cell class, you see differences. And so one thing I want to sort of highlight is, so cells, for example, D and F, they send their dendrites to this middle part of the retina, which is really different from cells I and G, which send their dendrites to the top part of the retina. And from this, we can already start to guess that they're getting input from different cells. So I and G are getting input from cells with axons up here, and D and F are getting input from cells with axons in the middle here. So this brings me to why we care about morphology. As I sort of just referenced on the previous slide, we can hypothesize as to who is providing input and which cells are receiving the input from our cells of interest just by the morphology of a neuron. And here in each gray box, I'm showing a different retinal cell class to also illustrate the point that we can tell whether a cell is excitatory or inhibitory based on its morphological features, for a given brain region. So in cortex, we know that excitatory cells have really spiny dendrites, whereas in the retina, we know that inhibitory cells send their information horizontally. So hopefully you can already guess that these horizontal cells are inhibitory, but also amacrine cells fall into that category. And we also know that excitatory cells send their information vertically. So that includes the photoreceptors, bipolar cells, and retinal ganglion cells. And so already we can start to have a sense of how these cells are connected to others, what kind of information they're carrying, whether it's excitatory or inhibitory, just from the morphology of the neurons. And also, what the identity of those cells are by looking at that morphology, because within a cell class, there is variability, but you can tell an amacrine cell from a bipolar cell. And so next, I'm going to talk about the neuroanatomy of primary visual cortex. So leaving the retina now, going to primary visual cortex. And I want to focus on this structure in large part because this lesson plan looks at neurons from cortex, primary visual cortex of mouse, or other human cortical regions. And so I want to give a brief overview of the neuroanatomy of this structure because it can be a really important point for the lesson plan. So mammalian cortex typically contains 6 layers. I'm outlining them here. Layer 1 is at the top by the peel surface, and then they're numbered sequentially down with layer 6 being at the bottom here. Now this is a schematic of mouse primary visual cortex. And typically in mouse, we group layers 2 and 3 together and actually split layer 6 to a part A and part B. But we care about these cortical layers in part because we know that information flows differently through the cortex depending on these layers. So information arrives at primary visual cortex into layer 4. Layer 4 neurons send that information up to layer 2-3 neurons. Layer 2-3 sends that information down to layer 5. And then layer 5 neurons send that information to other cortical or subcortical regions. So already we know that neurons in different layers can be carrying different information and sending their information to different locations. And so hopefully you can already have a sense that those neurons might have different morphologies. We also know that the cortical layers are formed at different times, so in an inside-out manner, meaning during development, layer 6 neurons are generated first, then layer 5, 4, and then so on and so forth to layer 1. And this is important to know because we also see that there are differences in the cell composition of those cortical layers, which are generated at different times. So if I click here, and you can now see an actual image of the primary visual cortex of the mouse with those layer boundaries still present. Hopefully you can see in layer 1, there's not a lot of dots here indicating not a lot of somas, whereas layer 4 is very dense and has a lot of cell somas. And so we know that cortical layers contain distinct numbers and types of neurons. And as I said, they can have really different morphologies. And so students will be able to compare neurons from the same species, but across layers, or within the same layer, but across species. And that is important for part 1, but especially part 2 of the lesson plan. And now I will hand it over to Stacy to talk about cell types.
Thanks, Claire. I'm Dr. Stacy Sorensen. I am the Associate Director of Neuroanatomy at the Allen Institute for Brain Science. Today, I'm going to be talking to you about cell types and in particular what they are and why they are important. Scientists have been interested in cell types for a very long time. As Claire mentioned, cells with different morphologies are frequently grouped into different cell types or classes. But what we really want to understand about cell types is their function, but that's often difficult to determine in the brain, particularly for complex structures like the cortex. Luckily, there are many more ways to characterize cells. Morphology is an important one. Cells with the same morphology are frequently grouped into a cell type. But there are also other important cell features such as electrophysiology and gene expression that can be used to characterize cell types. Each of these features gives us important clues about the function of a cell. And then each of these properties can be used alone to describe cell types. However, I want to tell you about how we can use them in combination to generate a more comprehensive, robust description of cell types so that when students start going through this lesson plan, they understand the morphology. That's a powerful cell type descriptor. It is most powerful when it's studied in combination with other cellular properties. So why are we interested in cell types? As I mentioned, they tell us something about the function of individual neurons in the brain. But cell types also give us this parts list of the functional cell types. They're thought of as the fundamental building blocks of this complex organ, which consists of close to 100 billion neurons in the human brain and 100 million neurons in the mouse brain, which is the size of maybe an eraser. In order to understand the whole brain, we need to understand all of its parts. Though our current focus at the Allen Institute is to understand cell types in the normal brain, there are numerous diseases that are known to affect specific cell types. Some examples are shown here. We've got Huntington's disease, Alzheimer's disease, but once we understand them in a normal brain, understand cell types in a normal brain, we may be able to better understand how they are altered in diseases like Huntington's, and to identify ways to alter them through gene therapeutics or even to compensate for their loss. But our first goal is to identify and characterize cell types in the normal brain so that we have that foundation for future work on disease models. Though the focus of the lesson plan is morphology, an extremely valuable new method for describing cell types has also emerged in the neuroscience field recently and beyond. It's called single-cell transcriptomics. With this method, we can describe the complete gene expression profile or the transcriptome of thousands of dissociated individual neurons. In this case, we're interested in two mouse cortical brain regions, ALM and VIS-P, which can be dissected from sections of the mouse brain and processed for transcriptomic analysis. And that's illustrated here. Using this and similar methods, the transcriptomes of millions of individual cells have been characterized. We then use these transcriptomes to identify cell types based on the pattern of differentially expressed genes. Here I have cells grouped into a hierarchy where cells are split out from glial cells. You see neurons here and astrocytes, oligodendrocytes over here, and then further broken down into major classes of GABAergic and glutamatergic neurons, and then into subclasses such as P-valve or parvalbumin or SST somatostatin. And then the individual types are shown down here in these rainbow colored names. These groupings of cells give us a taxonomy of transcriptomic types or T-types. Since this taxonomy is built using very large numbers of neurons, it provides a very stable framework to use for understanding cell types. However, a major question for us is, how do these transcriptomic types relate to cell types described using more traditional methods that measure morphology and electrophysiological properties? So in a parallel study at the Allen Institute, we used the patch clamp method to make electrical recordings from visual cortical neurons in acute brain slices kept alive in a dish. Here, this glass electrode is the way that we talk to cells using electrical impulses. And that allows us to describe their response properties in response to electrical stimuli, and to fill the cells with dye, which later we can, so that's an example of one of the electrical responses that we're measuring and what the individual responses look like and the types of things we can measure from it. And then when we fill the cells with dye, we also get the morphology. First, we have to take a picture of the neuron throughout its depth, and then we can create a drawing or a tracing or a 3D reconstruction of the neuron, which gives you a representation like you see on the right here. And this is, what we'll be teaching you to do over the course of this lesson plan, you or your students to do. Okay, so we then use these data to the morphoelectric properties that we measured from individual cells using our patch clamp method. We then use these data to establish a taxonomy of morphoelectric types shown here in mouse visual cortex. And you can see that each type has a neuron with a different shape shown up top, and then a different electrical response, as you can see down below. But we still had a problem. How do we connect these two taxonomies? Luckily, we have a method called PatchSeq, which was developed by our collaborators Andreas Tilius and Sten Linnerson, which essentially merges the transcriptomics data generation pipeline with that patch clamp data generation pipeline. And then to get the genetic information from each individual neuron, we suck out the nucleus shown here in red, and then some of the cytosol seen in blue, and send that for sequencing for DNA sequencing. And then we map these sequences to the existing transcriptomic taxonomy that I showed you previously. And then we can directly connect T types to these more traditional ME or morphoelectric descriptions of cell types. So we think of it as putting flesh on the bones of the transcriptomic taxonomy. So just that last graphic showing you how we take that single nucleus and then get the transcriptomics in addition to the morphological and electrophysiological properties from each neuron. Okay, and then here I'm just showing an example of morphoelectric transcriptomic type taxonomy or MET type taxonomy. We see many of the cell types that had previously been described by other studies, but we also describe additional diversity, for example, within the SST type seen here, which formerly had just about 5 different cell types described for cortex, and also for parvalbumin neurons over here in red, which there were about two of those types previously described, 2 or 3. And then one of the reasons we started with inhibitory cortical neurons is because their morphology is much more conserved in this acute slice preparation that we use for these studies. And as we move to describing excitatory or glutamatergic multimodal cell types, we know we're going to be missing a big part of the equation, since these neurons have long range axons that travel across the entire brain, and which will get cut off in these acute slice experiments. But we have a way that I'll tell you about to bring those data back into the mix. So in our current studies, we're starting to use the local dendritic morphology of excitatory MET types to connect to full morphology data collected through a separate data generation pipeline. And here I'm showing the local morphology of a neuron collected with PatchSeq on the left, with the apical dendrite in light blue and the basal dendrites in dark blue, and the local axon in gray. And it's predicted long-range projections by mapping to this cell over here, which we reconstructed from a whole brain image. And the local morphology is very similar between the two. So here I just wanted to show you the set of excitatory MET types that we identified. and how we've been able to use dendritic properties to connect to neurons labeled and reconstructed in the intact brain so that we can make predictions about the relationship between gene expression, EFIS, local morphology, and long-range axonal projection targets. And then I'm showing each of these data modalities here. So just to summarize what I have talked about today, we have established methods for classifying neurons into multimodal MET types or morphoelectric transcriptomic types. We've defined a comprehensive set of MET types for a single brain region, visual cortex in the mouse. We've built methods to use local morphologies to map cells across data generation platforms, full brain morphology, nanoscale local synaptic connectivity, to build richer definitions of cell types and to describe the relationship among different cellular properties. These methods are being scaled up to cover the whole brain across species, and then we'll do this starting with transcriptomics, and then we'll add in additional modalities like morphology by utilizing PatchSeq and other methods. Through transcriptomics, we have a fantastic way to connect cell types across species, even though the gross anatomy of the brains are very, very different, which gives us access to cellular properties that we can't always access in human tissue samples, for example. And it deepens our understanding of how the human brain is organized. Thank you for listening, and thank you for your interest in morphology and cell types. And I will hand it over to Lauren.
All right, thanks, Stacy. I'm Lauren Filer, and I'm going to be talking to you guys about axon, dendrites, and soma. I'm a research associate here at the Allen Institute, and I help kind of mainly wrote the lesson plan you guys are hopefully using to teach your students. So I want to kind of take a step back and talk about the inspiration for the lesson plan. So if you've taken any chemistry, even simple chemistry, you're probably really familiar with this model of an atom. You know, it's very simple with its nice nucleus, with the protons and neutrons and electrons orbiting in that very classic shape. But as you advance more in chemistry, you learn that this model of the atom is quite inaccurate. In fact, it's more of an electron cloud. And that model helps you realize, you know, kind of a more accurate impression of what an atom actually looks like. Unfortunately for neuron morphology, this is an image of neuron that's taught, you know, all the way back to, you know, middle school or high school intro biology class, all the way to senior level undergraduate college degrees in neuroscience. In fact, this image was from my college textbook about neuron morphology. And here is what a neuron actually looks like. Here it's in an image reconstructed here at the Allen Institute of a full neuron across a mouse brain. And you can just see how much more complex that neuron it is. And the reason why this is important is because when students are only thinking of neurons looking like this third image, it gives an inaccurate impression of the scale, complexity, and diversity of neurons. In fact, personal anecdote for me and most of my friends, when I started showing them the images I reconstructed, they're like, whoa, I didn't realize neurons look like that, because, you know, although they have degrees in neuroscience, they studied something that was not particular neuron morphology. So we realized, you know, if we have college level people with degrees in neuroscience not realizing what neuron morphology looks like, we wanted to make sure that it gives a more accurate impression like you would learn. So to start out, we're going to talk about what's mentioned in page 9 of the lesson plan, where we talk about dendrites. Stacy might mention the Ramon Cajal is a very distinct, you know, forefather in neuroanatomy, and he often drew the dendrites because they were the start of neurons. It's also because they were very distinct. They had these huge, large diversity in the shape and the structure of these neurons. Now, the shapes of these dendrites is actually not just willy-nilly. It's actually really regulated by proteins and other external factors. And such of these factors are the endoplasmic reticulum, or ER, as your students may be very familiar with them. And because science is still evolving, we are actually still trying to study how the shape with the ER is determined for dendrites. But because it's so regulated, we know it's a good factor in what we use to look at for cell typing. Now, a particular type of dendrite is called apical dendrite, and we show briefly in page 7 of the lesson plan. The reason why apical dendrites are so special and has such a call out is because they're a distinct feature in a certain type of cell called pyramidal cell. Pyramidal cells are excitatory cells. In the lesson plan part 2, we will go a little bit more in depth about excitatory versus inhibitory cells, but I just want to get a moment to talk about apical dendrites and its context in pyramidal cells. So pyramidal cells are found throughout the brain. It has not only the cortex, but hippocampus, memory center, or amygdala, the emotional center. But in the cortex, their pyramidal cells make up 70% of all neurons. And because they're so wide-reaching across the neuron, it's also, their features are well known. They're very thick. They reach towards that cortex layer 1 that Claire had mentioned earlier. And because of that, it's well studied, and your students might be interested in the fact that it's studied using stress, epilepsy, and things such as neuroplasticity. And with its studying, though, it's just has huge variance. This image right here is from Stacy's paper back in 2019. You can see how much the common featuring for most of these cells of this long branch reaching towards layer 1. But you don't have to have a degree in neuroscience to see how different these apical dendrites look like. And in fact, when you look at layer 6B down here, these cells that start on layer 6B, their apical dendrite looks very more atypical. They're upside down, they're sideways. In the lesson plan, we have students, look at cell morphology and filter by cell type, layers. And if they should notice if they're filtering by layer 6B, their cells and their apical dendrites going to look very different compared to a cell that's in layer 1 or layer 2, 3, or layer 5. Something to know for your students that the layer 6B apical dendrites are very different. It's quite a way to document how much morphology is changed by soma location across the layers. Now, next we're going to talk about spines. Spines are mentioned in both page 9 and 16 of the lesson plan. Spines are the source of synaptic connections. And you can see right here that some cells are aspiny, meaning they're very thin with not many bumps, or some of them are spiny. They're very bumpy. In fact, and these bumps are well documented and well studied. So well studied that they have these actually funny names such as thin, stubby, mushroom, and cup. The reason why they're well studied is because spines are the source of synaptic connection. If your students learn about action potential or postsynaptic density, about vesicles having neurotransmitters such as dopamine and serotonin and being released to ion channels, they may be very familiar with this image. Now, the postsynaptic density is that spine, the spines we see on morphology. And that upper level is that axon bouton. So connecting that this image isn't just a one-off, it is actually part of the morphology, is a very important point to bring across towards your students, that it's all still connected. And here on the image on the right is a real reconstruction of a neuron. I think actually this image does a really good job of capturing what it actually looks like. You can see this lawn and kind of this pink color, this dendrite spine connecting to the axon bouton. And right there is where all your vesicles will be setting up the neurotransmitters between the two. Now, if we go upwards and we talk about the axon, the reason why axon is also mentioned is it's very different from dendrite. It's much thinner, it's much more wirely compared to a dendrite. On this image that I just propped up is an image of a dendrite branching schematic, or dendrite branching. You can see how simple it is. It goes 1, 2, 2, 3, 3, 4, 4, 4, 5, 5. But compared to the red in this image of the axon, it's very thin, it's kind of chaotic. You can't do that simple 1 to 2 to 2, 3. These are one of the features we use to help distinguish not only between dendrites and axons, but axons with axons, of like how chaotic the branching pattern is. And you still got to remember that because of this, some cells reach across the entire brain. So how far the axon reaches across the brain is another feature we use for cell typing. And lastly, I want to talk about soma. It's mentioned in page 10 of the lesson plan. Although the Allen Institute doesn't really study somas too much in depth in context of neuromorphology, it was very important for us to talk about it. So if your students have taken some cell biology, they're probably very familiar with this image. You have that simple nucleus in the center, you have the Golgi, you have the ER, you have the mitochondria. But you got to remember, a neuron is a brain cell. So all those organelles that are taught in standard cell biology are still found in the soma of a neuron, because in the end of the day, a neuron is a brain cell. And in fact, here is a EM or electron microscope image of a neuron, and you can actually see distinctly the ER right there, the nucleus, the mitochondria. And it's a good point to hit to your students. The reason why you're kind of taught the standard schematic of a cell that's kind of not really tied to any particular cell across the body, but it's because those organelles are shown consistently across all cells of the body, whether it be the brain cells, a neuron, a lung cell, a liver cell. But it's still all very connected. And lastly, I just wanted to end on this image about the density of neurons. Sometimes we often think of these neurons in isolation, but here is a live reconstruction of all the neurons in about, you can see the scale bar about, 450 microns, with 150 micron equal about two human hairs in diameter. And you can just see how compact these neurons are. And that these neurons are not just isolated, but they all work intricately together. And the question is, how do we quantify all these neurons for cell typing? And with that, I lead it to Alice.
Hello, my name is Alice McClure, and I'm a data analyst here at the Allen Institute. In the lesson plan, you learn about the quantification of morphology. And from our research perspective, I want to talk a little bit about what that means. I like to think of the quantification of morphology as the process of taking qualitative assessments and turning them into quantitative measurements. By that, I mean finding a way to define and measure features of a cell, a process we call feature extraction or feature calculation. This allows for a systematic, objective comparison of cells, both when looking at them individually or as a part of a group. Here are two sets of cells from a recent paper, grouped according to their transcriptomic type described earlier. I would like to note that these are human cells and not mouse cells, which you have seen earlier in the webinar. Just by looking at these cells, what are some of the ways we can describe the differences in their shape? The cells differ when it comes to size, depth, complexity, and a diversity of other metrics. But how do we define those differences as an aspect of a cell? When talking about size, we can talk about how long a cell is from top to bottom, or along the y-axis. We can also talk about how wide a cell is from side to side, or along the x-axis. And these are measurements that we can take. We can also find ways to define and elaborate on what makes a cell complex. One way we do that is by counting how many branch points a cell has, another measurement we can take and that Lauren noted earlier. By taking these measurements, we can now compare the morphologies of cells of different transcriptomic types and identify trends using box plots. These box plots allow us to see the distribution of a feature measurement for each transcriptomic type and perhaps make observations or conclusions. For example, the neurons in the bottom group, COL22A1, have sparser apical branching than the neurons in the top group, GLP2R neurons. We can see that in that they tend to have longer apical dendrites, as noted by their trend to be higher when we measure the apical maxillocutein distance, but roughly the same number of branch points, which we can see when we compare the number of branch points we see for the apical dendrite. In order to assess if these differences are significant, we use different statistical methods. While we can observe trends using these box plots, it's also worth noting that we can see a diversity in cell shape even within these two groups of cells. This can mean that even within a T-type, these feature measurements can vary widely. One solution that we introduced in section 2 of the lesson plan is to calculate something we call a histogram. Histograms are a way to represent patterns in the structure of all cells within a group instead of a collection of individual measurements. To calculate these histograms, we split the cortex into small regions called bins from the most superficial part of the cortex, the pia, to the deepest part of the cortex, the white matter. Both of these parts of the brain are present both in human cells and in mouse cells. Then we calculate how much of each cell within the group resides in that portion of cortical space, those small vertical jumps with those defined 5-micron boundaries. We then plot it relative to the cortical layers. In these plots, the y-axis denotes the depth within the brain from the pia to the white matter, as I described earlier, and the x-axis represents the total amount found within that bin. This allows us to summarize how a cell or a group of cells is distributed along the depth of the cortex, which gives us valuable information about where these neurons tend to send and receive information relative to depth within the brain.
Hello, everyone. My name is Grace Williams. I'm part of the morphology team at the Allen Institute for Brain Science. I'll be showing you how you and your students can reconstruct your own neurons. This is Mosaic Brain Builder, which you can find at mosaic.science. It is a program that allows people from across the world to learn about neurons and to further the study of neuronal cell types. For this, you can refer to section 1 of the lesson plan, which starts on page 6. As you can see, the Mosaic website has a number of features. Here you can learn about neuroanatomy and about the Mosaic project. You can also participate in community forums, and as I'll show you later, you can look at the final product of previously traced neurons. For now, let's jump right in and get started tracing. Before we get started, you may ask, why are we doing this? Why do we trace neurons ourselves instead of using the computer? Why do we gamify reconstruction like this? The short answer is that we just don't have the technology to capture such intricate shapes. We gamify reconstruction to get as much data as we can while educating people at the same time. So let's get started. This is the neuron we'll be helping with today. There's already a good amount of tracing done by others, so we're going to add on to that existing trace. This pink trace is disconnected and this light blue is connected back to the soma. So here's the soma right here. You can see a bunch of dendrites, which are thicker and spinier. And you can see some axon right here, which is this thin, smooth branch. So we're going to come over here. We can move from 2D image to a 3D image stack by toggling 2D, 3D over here in the toolbar. And we can zoom in by scrolling. Over here you can see the view controls. You can right-click and drag to rotate and shift right-click to pan. So we're going to add onto the branch right here. You can see that the reconstruction itself is essentially a series of nodes that are connected to represent dendrites and axons. So we're going to come over here to virtual finger. We're going to click on this node right here because we're going to be extending this branch. So you can see now we have it in the 3D space. I'm going to move a little closer using left scroll to go in. And then we're just going to click and drag as though we're drawing on paper. Follow the branch. And then you have some trace. Sometimes you'll have errors, which you can see over here. We've got a bit of trace that hopped off of the branch. So we're going to come over here to the toolbar and we're going to select delete node. So we're going to delete the nodes on either side of this error. And then we're going to delete that whole branch like this. There we go. And now I'm going to go back to virtual finger and connect those. Sometimes you might have to do that a couple times, but that's all right. You can actually come down here and use Magic Tracer, which will help you connect two nodes entirely. That's not true. Moving on. Okay, so I'm going to connect that back here. Do that again. There we go. We've got a nice straight branch going. Okay. Scroll out a little bit. Use virtual finger again. Now we are recentering over there. So I'm going to trace a little bit more and then I'll show you some more tools that you can use. And as I go along, I'm racking up some points here. check out the leaderboard later. Okay, so I want to start on this branch, but it's a little bit hard to see. So I'm going to come over here and I'm going to use the contrast bar so that I can see it a little better. And I'm going to go back to that virtual finger, select this node right here, and continue on the way I would the different branch. So this is going to be a little bit more difficult. So another tool that we can use is the Connect the Dots tool. And this restricts the volume to a single plane that we can scroll through. And there we can see exactly where we want to put these nodes. And that's what I'm going to do. It's a bit more precise than the virtual finger. And it puts things exactly where you want. Now if we go back to virtual finger, you can see that I've got that branch. Another way that you can trace is by using the Connect the Dots 3D. And that's similar to the regular Connect the Dots, but you just put the nodes in the 3D image where you want them. And that will also allow you to trace. And it makes things a lot easier when you have branches that are maybe a little more twisted or thin or a little bit harder to see. And that'll get you where you want to go. So there are other areas that you can contribute to. You don't just have to add branches on there. You can go over to other parts of the reconstruction and confirm previous reconstruction points that have been started by others. There we go. So here, this is a possible connection right here. And that looks pretty good to me. So I'm going to use my virtual finger. I'm going to connect those. And it's things like this that allow us to make a consensus among multiple players. There are some other tools that you can use. As you can see over here, you can, do a little more specific work using these settings, and that's all found in the Mosaic tutorial that you can get into in more depth. But for now, we've reconstructed some neuron, and you can see it's quite a lot. And that's part of why we gamify reconstruction so that we can, capture these great amounts of data. And there you have it. We thank you for all of your contributions, and we hope that we can be of help to you when you teach your students. Every node counts.
Thank you, Grace. Hi, I'm Julie Andrade Wilson, and I'm a research associate in the Neuroanatomy team at the Allen Institute for Brain Science. Today, I will be showing you how to navigate the Allen Cell Type database, which is introduced in section 1 of the lesson plan and features an extensive data set of morphological and electrophysiological characteristics of human and mouse cells. This tool allows for the exploration of cell properties and biological features from single human and mouse cells. To access the database, click the link on page 16 of the lesson plan or access through the brainmap.org website. On the top menu of the website, to the right of Allen Brain Map, hover over Analysis and Data, then click on Search Cell Feature Data. The database can be filtered for a variety of results. On pages 18 through 20 of the lesson plan, students will be prompted to select different filters to visualize specific data to answer the questions. they will use the species filter to compare mouse and human cells. This filter allows them to access exclusively mouse or human cells by selecting the box for the corresponding species. Alternatively, they can select both boxes to view cells from both species. The location filter features drop-down menus that allows them to select specific brain regions and/or cortical layers to filter for cells with those specific attributes. Note that it is possible to select multiple options from the same drop-down menu. Students will also be prompted to use the morphology annotation filter, which has 3 sections where they can select multiple types of morphologies to view. Check the corresponding boxes to filter out results from different reconstruction type, dendrite type, and apical dendrite status. This will allow students to view cells based on the type of reconstruction, presence of apical dendrite, and excitatory or inhibitory nature, which will be especially relevant for answering the questions in the lesson plan. Students can also click on the green Clear Filters button to quickly clear selections and start over with the new selections to answer different questions. The filter results will be displayed in cell cards containing the corresponding cell types data selected in the filters. Each card contains information regarding the cell and the donor's characteristics. Cards may also contain images of the cell's reconstruction. By clicking on the Morphology button, a new window will populate with more specific morphological information about the cell. On this new window, there will be a summary of the morphological characteristics as well as 2 images. On the left is the biosite and stain of the cell, and on the right is this 3-dimensional morphological reconstruction where you can see the trace axon and dendrites. Thank you, and I will be handing off to Rachel Daly now.
Hello, I'm Rachel Daly from the Neuroanatomy team at the Allen Institute for Brain Science. I'm going to show you how to navigate the mouse visual cortex patch-seq database, which is discussed starting on page 24 of the lesson plan. This is different from the database Julia just showed you, in that these cells have 3 data types available instead of just the 2. The data types include a cell's gene expression profile or transcriptomic type, electrophysiology, and morphology. From the main landing page, portal.brain-map.org, you'll click Browse Data, then you'll select within the filter box and filter by species mouse, technique, patch-seq. You'll select outside of the filter box and then click Browse Specimens. Now that we are looking at the data, we can see each row is an individual cell with its summary data at left, followed by morphology histograms, which Alice discussed earlier, electrophysiology plots, transcriptomic assignment, the thumbnail view of the morphology reconstruction, the MET type assignment, where cells with similar morphological, electrophysiological, and transcriptomic features are grouped together, and finally, another electrophysiology property, the action potential trace. You can edit your view options seen on the landing page by selecting the gear icon at the left. We'll remove response at Riobase and Riobase hyperpolarizing sweeps to simplify what we see below. You can edit the properties by which you can filter by selecting the gear icon in the upper right-hand corner. This was formerly done via Change Properties as seen in the lesson plan, but the website has since been updated. You can add features of interest. In this case, we'll add morphology available. Then you can click anywhere in the horizontal filter box near the top of the website, which will display a menu allowing us to filter by various criteria. On page 28, students are asked to filter by morphology available. You'll notice There are only a subset of cells with morphologies since not all cells meet a certain quality criteria. Next, filter by layer. Here I've selected only cells in layer 1. Minimize the filter options by clicking in the filter box at the top again. Next, students will review the displayed cells and then select one example. They'll note the cell's brain region. Currently, everything in the database is from the mouse visual cortex. They'll note its specimen ID, which is located in the upper left-hand corner and is a 9 to 10 digit unique number. They'll also note the T-type, in this case SST Chottle, and MET type, which for this cell is SST MET type 1. The students will then sketch the cell they chose in the blank layer diagrams provided, being sure to match the relative location in each layer of axon, dendrite, and the soma. Dark blue is dendrite, bright blue is axon, and the orange dot is the soma. You'll notice for this cell, the soma is near the layer 1, 2, 3 border, and that a few branches of axon extend all the way to the top of layer 6A. The students will do this for all layers. They'll return to the filter box, unselect layer 1, and select the next layer. On page 29 of the lesson plan, they will reflect on differences they see between layers, with the end outcome being the observation that a neuron's shape can vary by layer. On page 30 of the lesson plan, the task is similar, but instead of filtering by layer, we'll filter by transcriptomic type or gene expression type. You'll unselect our previous layer choice and double check that the morphology filter available is still checked. We'll scroll over to the colorful wheel at left, where students will select one of the outermost circles or transcriptomic types. You'll see when you hover over one of the circles, information is displayed, including the type name and number of cells in that type. Students will pick one of the outermost circles from each of the subclasses: parvalbumin, SST, VIP, and LAMP5. In much the same way as the previous task, students will scroll through the options, pick their favorite looking cell of that type, and sketch a cell from a specific transcriptomic type from each subclass and reflect on the morphology differences when looking at cells from different subclasses. Here we are looking at a parvalbumin relin ITM2A cell with a soma in layer 6A. You can see the axon stays fairly local to the soma. For the last task, students will need to remove the transcriptomic type by clicking on the X. For this task, students will compare cells found in different layers from within the same transcriptomic type. First, we'll select a transcriptomic type found in the outermost level of the circle. It's a good idea to select a type with lots of cells to increase your chances of finding that type in multiple layers. Here I've chosen LAMP5 PLCH2 DOC5. Then we'll move over to the cortical layer options and select a specific layer. You'll see for this type there are options for cells in 4 different layers. Students will choose a cell in that layer to sketch, then move to a different layer. You can see when I go back and unselect the layer, the morphology available filter accidentally came unchecked. So we're going to fix that and then go back and select a new layer. After doing this for 3 different cortical layers, students will reflect on how cells belonging to the same transcriptomic type might look different depending on which layer the soma resides. And with that, students will be able to complete pages 24 through 33 of the lesson plan. We'd like to thank you for viewing, and especially like to thank all of our team members involved in this research and our Institute founder, Paul G. Allen, for his vision, encouragement, and support.